Can everyone see that?
Yep,
Great.
looks great.
Okay, so as Jeff mentioned, my name is Cameron Woodard. I'm I just finished my PhD working in Dr. Lynn Raymond's lab at the University of British Columbia up in Vancouver. And today I'll be talking about two of the kind of main projects that I worked on during my PhD, both of which were generally focused around the main theme of automating preclinical testing in mice and in mouse models of Huntington's disease by integrating behavioral tests directly into the mouse home cage. So the main focus of the research in my PhD lab, as Jeff mentioned, is Huntington's disease. I'm sure most of you have heard of Huntington's disease before, but just as a bit of a refresher, Huntington's disease is a neurodegenerative disorder. It's actually the most common genetic neurodegenerative disorder, and it's caused by a trinucleotide repeat expansion in the gene that encodes a protein called Huntington. The Huntington protein is pretty widely expressed, but it's most highly expressed in the brain, and its exact function is not completely known, but it seems to interact with a lot of synaptic proteins, intracellular trafficking proteins, and it seems to be pretty important generally for neuronal function and survival. So in the Huntington gene, there is a polymorphic tract of CAG repeats in the exon 1 that varies in length between individuals. And in the unaffected population, there are less than 35 of these CAG repeats. However, in those affected by Huntington's disease, this trinucleotide tract has expanded and contains 40 or more of these CAG repeats. And so Huntington's disease is characterized as a movement disorder, and it does have prominent motor symptoms, most notable of which is probably chorea, which is a type of dyskinesia. However, in addition, there are also psychiatric and cognitive symptoms of the disease. And this triad of symptoms typically emerges in middle age and progresses over the course of several decades. And currently, unfortunately, there are no therapies that are available that can slow or halt the progression of HD. So one of the most striking features of Huntington's disease is the relatively selective pattern of neurodegeneration. And the area of the brain that is most susceptible to degenerate in this disease is the striatum, which is a brain region that's associated with a variety of different functions, but probably most closely with things like motor function and reinforcement learning. And in late stage Huntington's disease, up to 90% of the medium spiny neurons, which is the main neuronal subtype, in the striatum can be lost. And you can see that kind of in this illustration here with the dramatic enlargement of the lateral ventricles, which are directly adjacent to the striatum. In addition to the striatum, there can also be loss of cortical gray and white matter, particularly in sensory motor regions that project down to the striatum, and other regions, including other basal ganglia nuclei like the globus pallidus, as well as the thalamus, can also degenerate, although they're typically affected to a much lesser extent. So one of the most important tools that has been used to study Huntington's disease are animal models, and the most popular animal for this purpose is the mouse. For a variety of reasons, they're easy to breed and easy to introduce genetic mutations. And so there's a variety of different mouse models of Huntington's disease that have been developed that express this kind of expanded CAG repeat in somewhat different ways. One of the most well-established mouse models is the YAP128 mouse, which was developed by Michael Hayden's lab. And this is a transgenic model that expresses the full-length human Huntington gene with 125 repeats on a yeast artificial chromosome construct that's been transgenically inserted into the mouse genome. And this model has been relatively well characterized in the literature, and we've used it in our lab for quite a few years. Another more recently developed model is the Q175FDN mouse, and this model uses a knock-in strategy to just express the expanded human Huntington exon 1 with around 200 CAG repeats in one copy of the native murine Huntington gene. So this model has somewhat better construct validity, which basically just means that it replicates the genetics of the disease as it's seen in humans a little bit more closely, where there's one kind of expanded allele and one that's within the more normal range. Both of these models, we use both of these models in our lab, and they're both bred on the same albino mouse background strain. And they both have a slowly progressive phenotype that recapitulates several features of the human disease, such as the selective pattern of neurodegeneration, as well as aggregation of Huntington protein, which is kind of one of the pathological hallmarks of the disease. However, it's important to note that these mouse models don't show the same extent of degeneration as is seen in humans with the disease. And they also don't show kind of the more characteristic movement symptoms that are often seen in HD patients. And so a lot of the time, these models are kind of more modeling pre-symptomatic or early symptomatic Huntington's disease. And often it's necessary to perform behavioral testing to kind of tease out the behavioral phenotypes and identify any sort of motor deficits or other deficits in these animals. So there's a variety of different behavioral tests that are pretty commonly used to assess phenotypes in these mice. One of the most popular is probably the rotarod test, where you place a mouse on top of a rotating rod and you see how long it can stay on for essentially until it falls off. Often it's accelerating, so the task gets kind of more difficult as it goes on. So this is a test of full body motor coordination and balance. Another test that's sometimes used, again, of kind of full body motor coordination is the balance beam test, where a mouse just has to traverse kind of from one end of an elevated beam to the other. And then people also frequently look at just things like locomotor activity levels in an open field test. So if you just place a mouse into an open arena, you can see how far they move over a certain period of time. And so there's a couple of really important things to note here with this type of behavioral testing. The first is that when you're testing these kinds of genetic models, the severity of the behavioral phenotype can vary a lot from mouse to mouse. So you might have an HD mouse that looks pretty much identical to your wild type animals, and you might have a wild type, or wild type just means non-genetically modified mouse that behaves pretty badly on some of these behavioral tasks. And so what this means is that in order to kind of properly power your results, it's often necessary to test pretty large experimental groups, which can be costly and time consuming. And another important thing to note here is that the phenotype that you see in HD mouse models is often very complex. So it's not just that they have some mild motor abnormalities, but they might also show physiological changes. So for example, YAC128 mice tend to have increased body weight as compared to their wild type counterparts. And they might also have things like affective phenotypes. So if you test them on tests of kind of depressive behavior or motivation, I know Jeff has looked at this a little bit, they can show some amotivational deficits, for example. And the danger with that when you're looking at some of these tests is that sometimes, I mean, for something like a rotarod, it's fundamentally dependent on the animal's motivation to stay on the rod. So, for example, if an animal doesn't find the punishment of falling off the rod to be particularly aversive, then they might just not really comply with the test and fall off willingly. So if you see a rotarod deficit in an animal, you don't necessarily know whether that's purely just a motor impairment or maybe it's due to something else. And so these are kind of factors that are just really important to keep in mind when you're looking at the behavior of these genetic models. So an alternative strategy that has gained popularity in more recent years, probably over the past 10 years or so, is the use of home cage behavioral testing devices. And I'm using this as kind of an umbrella term to describe any sort of automated testing system that mice can access from within their home cage 24 hours per day, and that collects data about them or performs some sort of test on them or intervention. And so there's a lot of different commercial and open source tools that are available for this purpose, and they vary widely in their functionality. So for example, you can do things like just track the activity of multiple mice within the home cage as they kind of move around different obstacles. This is a open source tool that's been developed for measuring feeding behavior, so how much food animals eat. This is a system that was published by our collaborator, Tim Murphy, for doing automated body weight measurements in mice. So you don't have to take them out and weigh them. It just does it all automatically. And in addition to these kind of more basic physiological measures, you can look also at more complex kind of operant behaviors in the home cage as well. So probably the most popular tool for doing that is, or commercially available tool at least, is the IntelliCage, which is a system that has 4 different operant corners, essentially, and mice can interact with nose poke ports at these different corners in order to retrieve water drops from these spouts. And this is the only source of water that they have, so they have to kind of interact with these different corners in order to get water. And you can kind of program different learning rules into a system like this. So, for example, maybe a mouse can only get a water drop at a corner when there's a certain color light that's illuminated above it, or you can do a kind of a spatial alternation task where maybe they have to go to different corners in a particular order in order to get water drops. So this is used sometimes to look at kind of cognitive phenotypes in mice. And there's also been some efforts to look at more motor phenotypes in the home cage as well. So this is a paper by the Ovechki lab where they integrated a joystick task into the rat home cage. And again, this is to get water drops. So the rat would just have to kind of manipulate this little joystick. So why would we want to do this type of home cage testing? Well, there's a few main reasons. First of all, these home cage systems usually involve the automation of some process that you would otherwise have to do manually. And by automating this, you're increasing the throughput of your experiments. So especially when you have to test larger experimental groups, this can be really, really valuable. And this kind of leads to the second point here, which is that if you don't have to do very much to leave these home cage systems running, you can leave them testing animals for long periods of time. And that allows you to collect kind of rich longitudinal behavioral data sets about your animals. And the final point here is that this basically just has to do with the fact that anytime you pick up a mouse, you remove it from its home cage, and maybe you put it into some sort of testing device, you have the potential to cause a physiological stress response in that animal. And that stress response can interfere with the behavior that you're trying to measure. And so the hope is that by integrating some of these behavioral tests into the home cage, you're minimizing the influence of these stress responses because the animal is able to essentially do the task in an environment that they're comfortable with, and they're able to kind of initiate it as they desire. So given all these advantages, when I started working in the Raymond lab, we had some discussions, and our collaborator, Tim Murphy, was working on developing a home cage system. And so we thought that maybe home cage testing could be a really useful way to assess the behavior of Huntington's disease model mice. So as I mentioned, I'm going to talk about hopefully two different home cage systems that I worked on today. I'll mostly just focus on this first one, and then hopefully I'll have a bit of time to get to the second one. And so this first one is called PyPaw, and the main goal of this system was to assess forelimb motor learning and the kinematics of forelimb movements in mice in their home cage. So you can get a general idea of what this system looks like here from this rendering. We have a regular mouse shoebox home cage that usually houses 2 to 4 animals. And then attached to one side of it, there's this small chamber that mice can access through a small entrance hole. And at the far end of the chamber, there is a lever that they can manipulate, and then there's a spout that delivers water drops. So taking a closer look at this chamber here, there's a few different kind of important components. First of all, prior to starting testing the animals, we implant a small RFID capsule subcutaneously on the back of their neck. So this is basically like the same thing that you would put in a pet, for example, if you were microchipping them. And what this enables us to do is that we can now group house animals in the cage, and then when a mouse enters into the chamber, they're detected by an RFID reader, so we can load the relevant parameters specifically for that animal and save away the data for that animal. We know which mouse is being tested at which time. So the lever itself is basically just a small steel rod that extends around a centimeter into the chamber, and it's movable on a horizontal axis, just on one axis, so basically parallel to the ground, so the animals can pull it towards themselves. And it's restricted to a range of 30 degrees by these 2 metal posts here. And the lever is coupled to a small motor, and the motor essentially just applies a force against the lever so that at any time it just returns back to its start position if it's not being manipulated. And the motor also has a rotary encoder attached to it. And so that basically just measures and records all the positions of the lever with really high resolution. So the way the task works is that in order to initiate a trial, the mouse has to climb into the chamber. They go to this nose poke port at the end, and they nose poke. And then following a short waiting period, a 1-second waiting period, there's an auditory cue that plays that indicates they can begin the trial. And at that point, they can pull the lever backwards to initiate the trial. And then after the end of the trial, depending on whether they met the criteria for success, which I'll get into in a minute, if they have, then there's a drop that is delivered from the spout, which is again just accessed through this nose poke port. So the duration of the time that they're performing the trial and kind of interacting with it, they basically just keep their snout inside the nose poke port. And then there's just a short timeout period, 5 seconds, and then they can initiate another trial again. And one other thing I wanted to mention is I know that in this rendering here, this all looks like it's transparent. It's actually not transparent. It's 3D printed from black filament, but the floor is actually made out of glass. And so we have a little camera that we can mount below that films up into the chamber. So that way we also have video recording of all the trials as well.
Cameron, sorry to interrupt. Could I ask a question?
Sure, yeah.
I wonder if you could address like using water as a reward versus food, and why you guys decided to go with water.
Yeah, so I guess a couple of things. So first of all, there is some literature showing that water restriction tends to be less stressful than food restriction in mice. So using water for rewards is pretty common in, especially in these kinds of home cage systems. I think it's easier to implement because you know, a food dispenser, there's a lot probably more moving parts as compared to a liquid dispenser where you just have to have like basically a single solenoid valve that just opens for a split second, lets a little bit of water through, and that's it. You don't need to have any sort of pellet dispenser. The other thing is that with food, from what I understand, I know food is commonly used in rats, but mice tend to satiate more quickly, I guess. They get full off the food more quickly. So you can't get them to do as many trials, I guess, whereas with the water, you have quite small drops. So I guess that's kind of an advantage of using water drops. What I will say also is that they do learn this task. They learn to kind of perform the task relatively quickly, and they tend to, they do lose a tiny bit of weight at first as they're kind of learning the task, but they tend to regain that weight pretty quickly, and they have, you know, enough access to the water.
Awesome, thanks.
Yeah, no problem. So when we think about kind of what this task is involving and what we're asking them to do, it's kind of a very unnatural thing for a mouse to do. Essentially, you know, you want them to go into a chamber, to nose poke at a port, and to simultaneously grasp a lever and pull it backwards. And we can't actually tell them explicitly what we want them to do at all. All we can do is kind of selectively reinforce different actions that they perform. And so in order to basically get them to perform this task, we have a training, kind of a multi-phase shaping or training paradigm that the animals have to go through. And in the first training phase, when we first put them in, they initially can get a water drop just for nose poking at the nose poke port on a fixed interval 15-minute schedule. So one drop every 15 minutes. And the way that they're essentially positioned when they nose poke at the port, they kind of sit back a little bit on their hind legs, and their left forelimb kind of rests on a little ledge, and then where the lever is positioned is directly beneath where their right forelimb is. So as they're nose poking to get water, they kind of naturally just grasp the lever, and through, you know, chance, they happen to move it a little bit and quickly discover that if they pull the lever anywhere past this kind of 3-degree threshold point, they'll receive an additional water drop. And so in this first training phase, any pulls of any amplitude, so it doesn't matter whether it's 3 degrees or 30 degrees, are rewarded. After they've done 100 rewarded trials in this first training phase, each mouse will be moved on to the second training phase, where they have to pull the lever a little bit further back. So now they have to pull it past the 8-degree point in order to get a water drop, and we also remove the nose poke rewards. After another 100 successful trials, in the second training phase, the mouse is automatically advanced to the main testing phase. And here the task gets quite a bit more difficult. So the main criteria for success in the task is the amplitude of their pull, so how far they pull the lever back. And essentially they have to pull it back to between 15 and 27 degrees in order to get a water drop. If they pull it back too far, so if they pull it to 30 degrees, they do not get a water drop. And similarly, if they pull it less than 15 degrees, they're also not rewarded. And we call those undershot and overshot trials, respectively. And then we also don't reward the trials if they're held away from the start position for longer than 2 seconds. So we just want to kind of reinforce them to do a short kind of ballistic backwards movement. And then if they just let go of the lever, it'll return back to the start position. So to give you kind of a better idea of what this actually looks like, I just have a couple videos here of mice performing the task. In this trial, you'll see, so just to orient you a little bit, I guess, we're filming from the bottom up, as I mentioned. This is the mouse's hind limbs. This is their body kind of extending through the chamber. And then their head is out of view. Their snout is kind of in the nose poke port, which is right here. And then this is the lever extending into the chamber. Their right forelimb is going to grasp the lever and pull it backwards. And then their left forelimb is just resting on this little ledge that I mentioned here. So what you'll see is the mouse grasps the lever, they pull it backwards, play that a couple times, and it goes just past the halfway point of the range, and then they let go of it, and it goes back to the start position. And so this trial was successful. It was rewarded. This trial here was, they didn't pull it quite as far back, so you'll see that it goes slightly not as far, and this trial was not rewarded. So I hope what you can see from this is that the difference between a rewarded trial and a not rewarded trial or a missed trial is very narrow. Actually, the reward range is only about 4 millimeters wide. So they have to be pretty precise in their performance of this task. So it's a relatively difficult motor task for them initially. So the first thing that we did was we just tested a cohort of young 2 to 3 month old wild type animals on this task. And because it's, as I said, relatively difficult at first, if we just look at the blue line here, this is their reward rate. Initially, they have a pretty low success rate on the task. Only around 20% of their trials on the first day are rewarded or meet the criteria for success. But we can see over the course of 1 week of testing, they reach around a 50% reward rate. And you can also see that the some of the failed trial categories, so the overshot trials, and then these are the held too long trials, decrease over this same period. If we test them out for a longer period of time, out to 3 weeks, you can see they do continue to improve. So by day 21, they reach around a 65% success rate on average. But the magnitude of the improvement from the end of week 1 to the end of week 3 is relatively small as compared to how much they improve in this first week. And so, and this is kind of in line with what we know about motor learning generally, where there tends to be kind of rapid initial gains in performance, then followed by longer and more incremental improvements beyond that initial learning. So for the rest of this, I'm just going to focus on this first week of testing, because this is kind of really when we seem to see the largest changes in their behavior. So what I'm showing you here is lever position versus time plots for 50 trials from a representative mouse on the first day of testing and then on the 7th day of testing. And so what you can see on the first day of testing is that the majority of their trials are overshot, or this mouse, for example, the majority of their trials are overshot. They also have some undershot trials, and then only very few, only like 1 or 2 of these trials are landing within this rewarded range, which is just indicated by the gray shaded region here. But by the 7th day of testing, they've substantially changed their behavior, and now they're terminating the majority of their pulls in this rewarded pull, or the rewarded position range. So if we look at this on a population level, this is just a probability density plot of pull amplitude. It's basically like a smooth histogram of pull amplitude from day 1 of testing in blue to day 7 of testing. And you can see that the distribution of their pull amplitudes shifts significantly over 1 week, so that a much greater proportion are now within that rewarded 15 to 27 degree range by the 7th day of testing, and there's fewer of them that are in this kind of undershot and overshot ranges. So something that's kind of interesting, actually, if you look at this distribution, you can see that the average on day 1 and the average on day 7 are actually pretty similar. So it's not actually that the average is really changing, but what's happening is the distribution is coming closer together. So it's kind of coming into the middle. And the way that we can quantify that is by looking at the intertrial variability of pull amplitude. So as I mentioned, if you just look at mean pull amplitude, you can see it doesn't really change across 7 days of testing. It's the average is within the reward range, even on the first day, and barely changes at all. But if you look here, on what I'm showing is the Fano factor of pull amplitude, which is basically just a measure of the intertrial variability. It's the variance divided by the mean for each daily bin. And you can see that this measure of intertrial variability decreases by about 40% over the course of 1 week of testing. And then what I'm showing you here is the relationship between the proportion of rewarded trials on each day for each mouse and the Fano factor of pull amplitude, the variability of pull amplitude. And what you can see is that there's a strong negative correlation between these measures. So essentially, on days when the animal has a reduced intertrial variability of pull amplitude, they have a higher reward rate. So what this kind of indicates is that animals are decreasing the variability of their movements, or the intertrial variability of their movements, over the course of 1 week in order to improve their performance of the task. So something else that we were interested in looking at was how trials were kind of distributed over the course of the day. And animals obviously have kind of a circadian pattern of their behavior normally. So if you plot out the distribution of the trials over the course of an individual day on average, you'll see that they tend to increase their activity in the last few hours of the light phase. So these mice are on a 12-hour light and then 12-hour dark cycle. And then they peak in their activity in the first few hours of the dark cycle before kind of starting to decrease a little bit into the early morning hours. And so this is generally, is in line with what we would suspect to happen in mice. Mice are primarily nocturnal animals, and they do have around, I think, 65 or 70% of their trials happening in the dark phase of the light cycle. I was also kind of interested in looking at how trials were distributed through the day on kind of a more local or short-term level. And so when I plotted out trials on just a line plot here, trials are just indicated by these blue lines. What I found was that animals tended to perform many trials at once. So they kind of clustered their trials into these short bouts of high task engagement that were then separated by longer periods when they didn't perform any trials. And as opposed to, performing their trials kind of more sparsely throughout the day, like, for example, going in and just doing 2 or 3 trials and then coming out and then going back in and doing another 2 or 3 trials. And so I used kind of an algorithm to cluster these trials together by looking at the average trial rate and then clustering them when the average trial rate went above a certain value. And using this strategy, I found that over 90% of the trials occurred during these short bouts of high task engagement, with the remainder of the trials showing kind of a more sparse distribution throughout the day. And these trial bouts were on average about 10 to 15 trials long, and mice tended to perform around 15 to 20 of these bouts of trials per day. So something I thought that was interesting when I looked at these trial bouts was that if I just looked at the reward rate or the success rate in the trials that were occurring within bouts, it was much higher on average than the success rate in trials that were more sparsely distributed or that were not in the bouts. And so one reason why this could be is that by clustering the trials together, the mice are actually able to have short-term improvements in their motor performance. And so when I looked on the level of individual bouts, so this is just looking at all bouts with at least 10 trials, and then looking at the reward rate over the bout, kind of in a 5-trial window, just moving over the course of the bout, what I found was that reward rate increased over the course of individual bouts. And at the same time, this measure of intertrial variability of their pulls, the Fano factor of pull amplitude, decreased over the course of individual bouts. So this kind of suggests that on both short time scales, so matter of minutes, as well as longer time scales over weeks, mice are able to kind of decrease the variability of their movements in order to improve their reward rate and get more rewards. Okay, so next we moved on to look at our Huntington's disease mouse models.
Just a moment, Cameron. There was actually a couple of questions that came into the chat.
Yeah, sure.
Did you want me to read them out loud?
Yeah, sure.
Okay, so one of them was, did you compare your trial bouts with the distribution of drinking under control conditions when they had access to free water?
No, I have not. So I haven't looked at. Yeah, so I suspect that the clustering into bouts is related to kind of the natural distribution of how they might drink normally. Like, I mean, 1 drop or 2 drops is probably not enough to like sate their thirst. So they might just want to get more drops in order to kind of then go and eat or something like that. So I haven't actually looked at that, but I think that's probably pretty likely that it has something to do with that.
And then there was just one more question. Is the clustering of bouts reflecting normal consumatory behavior?
Yeah, so yeah, I guess that's kind of similar to what I was just saying. That could be, yeah, that could have to do with just their consumatory behavior. But yeah, it would be interesting to actually see the comparison with how much they do tend to just drink normally, because so something else that I did saw, I'll get into this a little bit when I look at the Huntington's disease mouse models, but actually, if we look at some of the HD mice, their bouts tend to have the same length, even though they have a lower success rate. So they're getting fewer actual drops of water, but they're still making their bouts the same length. So that actually also makes me think that there could be a factor of fatigue as well. So maybe they just don't want to do more than 15 to 20 trials at a time because they get fatigued after doing that many, or it could just be, they kind of, that's their attention span for the task or something like that.
There was a follow-up question about the lever. Is there resistance on there, or can it be varied?
Yes. So there is basically the motor provides a fixed level of resistance during the trials itself. It's basically at the minimum level that we can set it for the motor, where it will reliably return the lever back to the start position afterwards. So we try to not have it at a resistance level where it's actually hard for them to pull, but we just want it to be basically, yeah, be enough to bring it back to the start position at the end of the trial. But that can be varied as well. And actually, there's another student who's working on the task in the lab now, and he's trying to do perturbations during trials. So, for example, the mouse pulls the lever backwards, and then the force on the lever changes slightly, and they have to adjust their movement in order to compensate for the difference in force. Okay, so looking at our HD mouse models, we found something kind of surprising, which was actually that in the YAC128 mice, and this is at a relatively symptomatic time point, so this is 10 months old, we found that the YAC128 mice actually had relatively normal motor learning on the task. They essentially learned the task at an equivalent rate to wild-type animals, but Q175 FDN mice had really severe and kind of pronounced motor learning impairments on this task. So even after 1 week, they only got to around a 20% reward rate on average. And when we took them out we did test these animals out to 2 or 3 weeks as well. And I think after 3 weeks, the Q175 still only got to around maybe 25 or 30% reward rate. So it seems like they were learning at a slower rate, and they also plateaued much lower. So their ultimate attainment was not as high as the other genotypes. This didn't seem to be due to baseline differences in performance. So if we just looked at the proportion of successful undershot, overshot trials on the very first day of testing, there was no genotype differences. So they looked pretty similar to each other on the first day. But by the 7th day, you can see that the Q175s have a greater proportion of specifically overshot trials as compared to the other genotypes, and of course, a lower success rate as well. So something else I was curious to look at was whether this failure to kind of learn the task as well was due to kind of overt differences in how they were pulling the lever. So I looked at a few different kinematic measures of their pulls. I looked at the average pull amplitude, the peak velocity of their pulls, and the peak acceleration of their pulls. And I didn't see any differences between the genotypes on any of these measures across a week of testing. So this suggested that the impairments were not really explained by differences in the mean kinematics of their movements. So the next thing I just looked at was the distribution of their pull amplitude again. And in the wild types here, this is the 10-month-old cohort, but you can basically see something similar to what we saw in the younger cohort, where there was a shift in their distribution of their trials over 1 week, or the distribution of their pull amplitudes on the trials over 1 week, so that there's a greater proportion of them in the rewarded range. If we look at the Q175s, though, there's much less of a shift in their distribution across 1 week of testing. And if anything, there's mostly just a shift to kind of higher pull amplitudes generally rather than to within the reward range specifically. So it doesn't seem like they're really learning that they have to kind of narrow in and decrease their variability onto this specific range of pull amplitude. So when I quantified the intertrial variability of their amplitude, their pull amplitude, I found that overall the Q175s had a higher Fano factor of pull amplitude across the full testing period as compared to the wild types. But there was actually no interaction effect here. So the Q175s did seem to decrease this variability to a similar extent to the wild types. So this would suggest kind of, at least on face value, that the Q175s were still able to decrease the variability of their movements to an extent, perhaps in order to increase their success rate. But when I looked at the relationship between this measure of intertrial variability of their movements and reward rate, again, in the wild types, I saw, again, each point here is just one mouse on one day. So in the wild types, again, I saw this inverse correlation where, higher reward rate on a specific day was associated with lower variability of pull amplitude, but there was no correlation in the Q175s. So basically what this indicates is that even though we're seeing a decrease on a group level in the Q175s, that's not correlated with improved performance on an individual level. So in other words, the mice that are decreasing the variability of their movements are not necessarily performing better, and the mice that are more variable are not necessarily performing worse. So it could be the case that the mice that have lower variability by day 7 are pulling more consistently, but maybe they're pulling consistently to the wrong place. They're pulling consistently overshot or consistently undershot. So they're just not really learning that they're supposed to pull to that kind of correct middle position range. So I also just looked at this over the course of individual bouts, as I had previously. And interestingly, I saw that while both wild type and Q175 mice did increase their reward rate over the course of individual bouts, only the wild type, but not the Q175, had a significant decrease in the variability of their pull amplitude over these individual bouts. And I know these look kind of similar, but you'll just have to trust me that the statistics bear out on that. So this suggests that maybe the Q175s might be employing kind of alternative strategies to improve their reward rate over the course of these individual bouts of trials, possibly by modulating their mean pull amplitude or something like that, but not by decreasing the variability of their movements. Okay, and so one other just behavioral thing that I was curious to look at was the circadian distribution of their trials. And one reason I was interested in this was because Huntington's disease patients often have sleep disturbances, and there's a higher prevalence of circadian sleep disorder as well. And so we were curious to see if we could see any differences in the circadian distribution of trials between our HD mice and wild types. And we did actually see that the Q175s and actually also the YAC128s, I didn't show it here, but both genotypes had a altered circadian distribution of their trials as compared to the wild types, although it was not exactly what we would have expected. In this case, it was the Q175s actually had a second peak in their activity kind of in the early morning hours, while the wild types were decreasing in their activity, and then they had less activity during the day overall. So that was kind of interesting that we could also see that aspect of this, even though we weren't specifically designing this to look at circadian abnormalities. Okay, so as I mentioned earlier, the extent of the degeneration that is usually seen in these HD mouse models is relatively minimal. So it's unlikely that the impaired learning of the task in Q175 mice is due to kind of overt neurodegeneration or loss of the neurons that's underlying the behavior. This said, HD mice, including the Q175s, do show alterations in the baseline activity and activity-dependent neuroplasticity of the medium spiny neurons that are in the striatum. And this can appear quite early on, and it can precede the onset of degeneration by many months. So we next wanted to see whether the motor learning deficit that we saw in the Q175 FDN mice was at all related to differences in corticostriatal connectivity. And to do this, we collected brain slices from animals that were either naive to the task or immediately following training on the task. And we did whole cell slice electrophysiology experiments. So the disadvantage of using this type of collecting brain slices approach is that obviously you're kind of randomly selecting neurons, and you don't know what neurons are specifically involved with performing the task, or whether those neurons are at all modulated by the task. However, we did have one thing that we could take advantage of, which is that this task is unilateral. So mice are only trained with their right forelimb. So what our reasoning was is that if we looked at naive animals, so animals that hadn't been trained, and we didn't see any interhemispheric differences between the left hemisphere and the right hemisphere in terms of the activity of these neurons, but then we looked at our trained animals and we did see hemispheric differences, we could have some certainty that those hemispheric differences had something to do with training on the task. So the first thing that we wanted to do was look at naive mice, and what we did was we measured spontaneous glutamatergic activity in the medium spiny neurons located in the dorsolateral striatum, which is an area, a subregion of the striatum that mostly receives projections from the motor cortex. And so we looked at the frequency of these spontaneous glutamatergic events as well as the amplitude, and we found that overall the Q175 FDN mice had a lower frequency of these spontaneous excitatory postsynaptic currents, but no difference in the amplitude of these currents. We also looked at PPR. So basically to do this, you just have a stimulating electrode, and then you give 2 pulses with varying interpulse intervals. And you can look at the ratio of the second pulse to the first pulse. And often a higher PPR is taken to indicate a lower initial probability of release. So if you have a PPR above 1, that means that on the first pulse you had a lower release than on the second pulse. And so what we saw was that at the lowest kind of interpulse interval in the Q175s, there was a higher PPR. So together with the decrease in glutamatergic event frequency, we interpreted this as meaning that there was basically a decrease in glutamatergic input to these dorsolateral striatum neurons, medium spiny neurons. And importantly here, there was no hemispheric differences on any of these measures. So on EPSC frequency, amplitude, nor in the PPR. I didn't split it up by hemisphere here, but there was no hemispheric differences either. So when we looked at the trained animals, we were specifically interested to see if we could see any hemispheric differences. And in terms of EPSC frequency, we did not see any hemispheric differences in either the wild type or the Q175 mice. But in terms of EPSC amplitude, we found that in the wild type animals, there was a significantly lower average EPSC amplitude in the left hemisphere as compared to the right hemisphere following training on this task. But we didn't see any differences between the hemispheres in the Q175s. So this suggests that possibly following forelimb kind of motor training, there's a hemisphere-specific decrease in the average amplitude of these glutamatergic currents, and we're able to see this essentially in the wild-type animals that are learning more on the task or performing better, but not in the Q175s that are not performing as well. So this suggests that perhaps this is the failure to learn the task is related to some sort of underlying deficit in neuroplasticity. So to try to bring this a little bit closer to the behavior itself, we wanted to see if this kind of average measure of glutamatergic activity in the contralateral striatum was at all related to any of our behavioral measures. So we essentially just tried to see if we'd see any correlation between this electrophysiological measure and our behavior. And what we found was that actually this average left hemisphere spontaneous event amplitude was significantly correlated with the variability of pull amplitude, the Fano factor pull amplitude on the last day of testing. So basically what that means is that animals that had a lower average left hemisphere glutamatergic current amplitude also had lower variability of their movements with relation to the task on the last day of testing, and vice versa. And when we split this up by genotype, what we saw was that this relationship was significant in the wild type animals, but there was actually no significant correlation in the Q175. So again, this suggests that the decrease in the amplitude of the glutamatergic currents that we're seeing in the wild type mice in the contralateral cortex might be related to the improved performance on the task, and the inability of the Q175 mice to effectively learn the task might be due to this unimpaired ability to show this kind of neuroplasticity. So in summary, we found that mice were able to effectively learn this task and improve their performance of the task over the course of 1 week by reducing the intertrial variability of their movements, specifically of pull amplitude. And this happened over both short timescales, so over just bouts of a few minutes, as well as over longer timescales, so several weeks. Q175 FDN HD mice had substantial motor learning deficits that were not explained by differences in baseline performance or in their mean kinematics of their pulls. But rather, these deficits seem to be due to Q175 FDN mice being unable to improve their performance of the task by reducing the intertrial variability of their movements and kind of narrowing in on that rewarded position range. We also found that task training was associated with reduced spontaneous glutamatergic event amplitude in dorsolateral striatum medium spiny neurons that were contralateral to the trained forelimb in wild-type mice, but not in the Q175 FDN mice. And similarly, we found that decreased SEPSC amplitude was correlated with pull variability in the wild types, but not the Q175s. And the final point is that this system is relatively affordable to build. It costs around $500 to build 1 cage. And we hope to have kind of full build instructions and all of the 3D printer parts and everything like that online at some point in the relatively near future. And it involves relatively minimal experimenter intervention and upkeep to keep it running. So I know that I'm running a little bit low on time, so I won't really spend much time going over this second system, but I'll just very briefly kind of talk about the purpose of this system. This second system is called PIDOS, and the goal here was not to actually assess the behavior of animals, but actually just to facilitate long-term drug treatment studies in mice. So the idea is the same. There's basically a chamber that's on one side of a mouse home cage, and the mice can enter into this chamber, and at the far end, there's no lever, but there's essentially just a spout, and they can lick at that spout, and the spout is connected to a capacitive sensor that detects the licks, and in response to a lick, it will deliver either a drop of water or a drop of drug solution. And so using the system, we can essentially administer drugs to animals without, in kind of an accurate way through oral administration. And there's also another important component here is there is a load cell. So actually the chamber itself is not directly mounted to the cage, but is just kind of sitting adjacent to the cage, and it's supported by this load cell. So when a mouse enters into the chamber and they're in range of the RFID reader, the load cell is collecting weight measurements from the animals, and then that way at midnight on each day, we can calculate an average body weight for the animal based on all those kind of load cell measurements and use that to update the drug dosage that the mouse receives. So this is just a little video of a mouse licking at the spout to get a drop. So this is just an example of kind of the body weight measurements that we can collect from the animals. Over the course of an individual day, we might collect 8,000 or 9,000 of these weight measurements from the mice, which corresponds to around 30 minutes in the chamber. We collect them at 5 hertz. And then at midnight on each day, we calculated an average body weight based on the mode or kind of the peak of this distribution of body weights. And just to ensure that this was an effective way to measure body weight, I had a cohort of mice that I put into the system, and I let them run for 2 weeks, and I manually measured their body weight each day and then compared it to the PIDOS calculated body weight. And I found that there was a really strong correlation between these 2 different weighing methods. So we're pretty sure that it's an effective way of weighing the animals. We also wanted to make sure that obviously the drug delivery was accurate and effective. And so in order to do this, as I mentioned before, for the water delivery for the PIPAW system, we basically just use a little solenoid valve that opens for a short period of time and delivers the water drop. But that's kind of prone to drifting over the course of days, especially as the volume of the water reservoir changes. So we wanted something that was a little more accurate for delivering these drug drops. And so we used this open source syringe pump design published by Winan et al. 2014. And we built 4 of these syringe pumps and found that they were very accurate. They had around 1% error for delivering these 10 microliter drug drops. So it seemed like a very accurate way to deliver drops to the animals. And we also just used a camera to basically confirmed that the mice were in fact drinking the drops that we gave to them. So it would take a picture of the spout immediately following delivery of the drop, and then we could double check whether the animal's mouth was on the spout, suggesting that they would have consumed the drop. Generally, the way that the dosing regimen works is that for an animal that is receiving a drug dosage, at midnight on each day, the system calculates their body weight. And then starting at midnight, if you just look at this middle panel here, they begin to receive drug drops. And then once they've received their full dosage of drug drops for the day, they're switched over to receiving water drops. So any subsequent licks at the spout will just deliver a regular water drop. And you can see also that over kind of a 2-month treatment period, the dosage of drug that they receive is scaled to match their body weight. So the dosage essentially remains stable over that full period. So in summary of this PIDOS project, although I didn't really talk about it too much, but basically this is a system to kind of orally deliver drugs to mice over long treatment periods in kind of an accurate fashion. It automatically weighs the animals and delivers drug solution using this open source syringe pump design. And mice pretty quickly learn to trigger delivery of the drug solution by licking at a spout, and we're able to maintain consistent dosing despite changes in body weight over the course of several months. And this system is actually fully available online in terms of the build instructions and all those kind of parts and stuff. We have an OSF page for it, as well as a Hackaday page that you can find linked in the paper. And so if you want to build one of these yourself to administer drugs to mice, you can do so. So with that, I'll just wrap up. I'd just like to thank my supervisor, Dr. Lynn Raymond, as well as our long-term collaborator on these projects, Dr. Tim Murphy, and then a number of members of the Raymond and Murphy lab, especially Maria, who have been really helpful in building these systems and working on all these projects. All right, thanks a lot.
Cameron, that's beautiful.
Thank you.
Really, really, really cool work. Questions from the Western crew? Oh, Janet has one, I'm sorry. Jenny, you want to just ask?
Sure. I just was wondering what your thoughts are as to why it seems the mice are persistent in overshooting the pull, and whether you can put that in a context in terms of how it might align with some of the motor symptoms in HD.
Yeah, so yeah, it seems kind of like what we're seeing here is that the Q175 mice are either performing a really short pull, so barely moving the lever at all, or they're doing a pull that's going basically all the way to the end, so overshooting, as you said. But they don't seem to really be able to kind of like effectively terminate their movements kind of mid movement. And there have been quite a few studies that have looked at reaching movements in HD patients and pre-symptomatic carriers. And they have found kind of impaired temporal sequencing of movement steps. So kind of the accelerative phase, decelerative phase, things like that, as well as kind of, there's one pretty big study that looked at error correction. So found that basically when there was little errors made early on in the movement, it was difficult to correct for those later on in the movement in HD patients. So I'm not exactly sure what this indicates, but it seems like there is a difficulty in kind of terminating ongoing movements, a reduced kind of endpoint precision. I don't think it necessarily has to do with feedback because it's very short. This is only 100 or 200 milliseconds long, so it's pretty short for there to be that much feedback. Yeah, I think it can be analogous maybe to some of the things that we see in the HD patients.
Thank you.
Thanks.
Thanks, Cameron. Other questions from folks?
Yeah, I had a question about kind of like your design process in designing PyPaw, and how many iterations, I guess, you had, and what were some of the big obstacles?
Yeah, so it was a very long process. What I will say is that actually the original home cage system was built by a couple of people in Tim Murphy's lab like 6 years ago. And at that point, it wasn't called PyPaw, and it was relatively different from the system as it was eventually became. But it still had the same general idea of basically having a lever, having a spout that delivers water drops, and animals kind of performing pulls to get water drops. But that original system we actually used to publish the first paper looking at Huntington's disease mice on this task. And the big difficulty with that was that there was no way to actually ensure that the mice were performing the task just with their forelimb. So you'd get mice that were kind of biting at the lever and doing the task just that way. So it's kind of difficult in these types of tasks just to make sure that the animal is doing it in a consistent way, in the way that you kind of want them to do the task. So after we kind of had published that first paper using the initial version of the system, I was kind of the main person that was working on it at that point. That was in 2017. And I realized I wanted to essentially kind of overhaul the system and change a lot of the things. So one of the things I did was add the nose poke port. I kind of changed the dimensions of the chamber. And I would say it took me like probably 6 months of, or maybe even more, of just working on the system and trying out different designs of the chamber and different protocols to actually get something that really worked. And there was a lot of iterations. I tried different orientations of the lever. One of the big things was also, I found that it was necessary to get the mouse a little bit in a more upright position. So if they're just down on all fours, it's kind of hard for them to grasp a lever and individually manipulate it with their forelimb, because they're already using one of their forelimb. They're kind of, you don't want them to be on kind of like a tripod position and then have the other forelimb kind of lifted up to pull. So I found it was better if we had them in kind of a sitting back, kind of more on their hind limb position. So I raised the ceiling up of the chamber a bit more, and then shortened it a little bit. And then that was, I found, a good way to actually get them to kind of reliably grasp the lever and pull it backwards, and also have enough room kind of between their body and where the lever started for them to actually perform a movement. So yeah, it was a long process of actually getting this to work before we were able to test animals. But then thankfully, once we did get it to work, we built like 8 of these cages, and we could test like tons of animals over the course of a year or two. So once it actually was working and we had the animals, it was relatively quick moving.
Cool, thank you.
Thanks.
Cameron, there was one more question from a student, Ellie, who said, is there a light from the upwards facing camera? If so, is there evidence of that acting as an aversive stimulus?
Yeah, so actually there's just a infrared light. So it shouldn't be, I don't think it's aversive. Yeah, it basically just allows us to film still at night, but it's very dull kind of red light. It's not particularly bright. So.
Oh, thank you.
Yeah, no problem.
Any other questions? I know we're butting up against people's dinner time / being outside in the beautiful Pacific Northwest time, but any other pressing questions for Cameron before we let him go? Cameron, I want to thank you again. This is so beautiful. This is like the future of preclinical behavioral research. I love the native home cage, the automation, the removing the experimenter error. It's really, really impressive work. And
thanks, Jeff.
We appreciate you guys giving us a talk.
Yeah, thank you, Cameron.
I appreciate you having me, and